A typical eukaryotic cell undergoes two main kinds of division. One is mitosis and another one is meiosis. Mitosis is known as a equational division, whereas the meiosis is a reductional division. We'll be studying in details about the second type, that is meiosis, in detail, but we'll also look into these few stages like G0, G1, S, and G2 phase. We'll also try to understand few other terms and certain concepts which we do not have much detailed idea about, like how does the spindle fiber it contracts and certain more important things. One of the most important part which we should know about is the C value. Basically, C value is the amount in picograms. When I say picograms, it means 10 to the power minus 15. And this generally means that the total amount of DNA contained within a sperm cell or a ova or any kind of haploid cell. But when we talk about a diploid cell, in that case, it is half of the total amount of DNA present inside a cell. So a cell in prophase has 2x, that is 2n number of DNA as a starting material. But this the forming daughter cells nuclei in the telophase cells will each have the same amount of DNA as the starting cell, that is 2n number, isn't it? And here this 2n number means that the chromosome numbers, when we talk about a chromosome number, we do not denote it on the basis of haploid number or diploid number. We denote it on the basis of n or 2n. So n number is present in case of only the sperm cell or ova or any reproductive cell, whereas 2n number is present in case of any kind of diploid cell. When mitosis is taking place in the prophase, also the cell is in 2n condition. When prophase is moving towards the end and it is attaining the telophase, although in the telophase what happens is, if you remember the structure of a cell, suppose there were three number of, I'm talking about mitosis. So suppose there were three number of chromosomes. As an end of telophase, we will have two cells from here which will be having three chromatids. Now here it has to be understood that this is also 2n, these two are also 2n. On the number of chromatids or centromere, we divide a cell, or we say that what is the chromosome content of that cell. So here also there is only one centromere, like three centromeres over here, three centromeres. So it is, in case of these places also there is presence of three centromeres, and that's why all these cells which are drawn over here, here this cell is at suppose metaphase, or you can think it to be just later stage of prophase. So similarly, suppose it's prophase, in prophase also it's 2n, and these all cells are also in telophase, but they are also having 2n number of cellular content. The nuclear content is 2n, chromosome number is 2n. This is to be understood. It is counted on the basis of chromatids. How many number of, not the chromatids, number of centromere. If the centromere number is the same, it means the chromosome content is also the same. This is about the C value. Now, what is the importance of C value? C values help in understanding that the cell needs to divide or not. Suppose C values have also been found to correlate inversely with cell division rate, meaning that large genomes are typically found in nuclei of large and slowly dividing cells. If the genome is very large, then the cell will require much more time to divide. So, depending on the genomic material, the cell division takes place. If the time period of cell division is determined on the basis of chromosomal content, because that is the substance which has to get duplicated, more the genomic content, more the time is required for the cell division to occur or to complete. Next is regarding all these different stages. When I say the different stages, it means we will be discussing from the G0 stage, although this is a stage which is not considered at all to be a stage. Then gap 1 phase, S phase, gap 2 phase, and these all are included in the interphase. I hope you all remember. And finally, our mitosis. Mitosis consists of all the stages of division like prophase, metaphase, anaphase, and telophase. Just do a quick recapitulation of these topics in order to understand the meiosis little more better. Because in meiosis also there are two steps. One is meiosis 1, another one is meiosis 2. In meiosis 1, actually the reductional division occurs. But in meiosis 2, it is the mitosis only taking place. So it is much more easier to understand if you can recapitulate the mitosis process. So we will be discussing about the condensation of chromosomes. We already know that inside a cell, when a cell is on the verge of getting divided, or just imagine that this is a chromosome. This is a cell having two chromosomes which are placed in the center of the cell in the equatorial position. Now what will happen? Now the spindle fibers will be released and the chromatids will be attached to one of the chromatid will move towards one side, another towards another side. How are the chromatids moving? The condensation of the spindle fiber is actually taking place. So how does this condensation of the like the spindle fibers occur? This is something we need to understand, and we are discussing it over here. Please try to have a clear idea about this. This is a mechanism that involves the condensing of the DNA that is synthesized during the interphase. The interphase stage allows DNA replication, which uniformly entangles or intertwines the DNA molecules for easy separation during mitosis. The chromosomes, they are getting separated. So this is what is getting explained over here. Chromosome condensation is the compacting. Compacting means summarizing or coming very close of the chromatin into well-defined rod-shaped structures known as chromatin. Here it should be chromatids. Okay, condensation allows structural reorganization to separate the identical sister chromatids, a mechanism known as a chromatid resolution. This is a biological term used for separation of the two sister chromatids which are held together with the help of centromere. Condensation forms sturdy elastic chromosomes. Sturdy, it means tough, but elastic chromosomes to prevent damage and breakage caused due to force which pulls the two chromatids apart with the help of these microtubules and the cytoplasmic pull which is experienced by the two chromatids, sister chromatids. Chromosome condensation is done with the help of condensins. These are certain proteins which help in breaking, like there are various kinds of protein. One is this condensin which help in condensation of the spindle fibers. There is separase which separates two of the sister chromatids which are intertwined or which are very closely placed to each other. So separase is also a very important enzyme which is used in separating two of the sister chromatids or breaking of the, suppose a centromere is there which is attaching two of the chromatids, sister chromatids together. When the sister chromatids divide, at the same time this centromere also divides. So if you always see the diagram in telophase, it's like this: two cells, both the cells have one of the chroma- uh, chromatid along with the centromere. So centromere is also getting divided. And how is it getting divided? With the help of this enzyme separase. That is also important. Condensins are complex proteins that play a key role in cell cycle and majorly in separation of the chromosomes. Here separase is not mentioned, but it is also there. Chromosome condensation, maintaining the structural integrity of chromosomes and the resolution of DNA topography. Topography is the structure during intertwining, that is entangling amongst each other. Topoisomerase is um, Topoisomerase is an enzyme which helps in providing stability to the structure which is newly formed. When two sister chromatids they are close together, it means they are definitely having some pull towards each other. Now when they are getting separated, so at that time this topoisomerase it binds around them and stabilizes both the structures. So topoisomerase basically helps in, specifically topoisomerase 2 helps in the relaxation and unlinking of supercoils formed in the interphase, cutting and releasing of the DNA duplex chromosomes. Condensation forms two sister chromatids which are egg-shaped and joined at a point known as a centromere. So basically, condensation of chromosomes is a process by which two of these sister chromatids they just form this particular structure, and the supercoiling of the DNA also takes place, and this structure is maintained. This is the condensation time where the spindle fiber is also um, learning how to condense with the help of these micro tubules and tubuline structures, as well as the structure of the chromosome is also becoming very prominent. Next is the movement of the chromosomes. This movement of chromosomes takes place with the help of like what is happening at the centromere are microtubules that function by recreating tubulins. Those tubulin molecules they start generating the spindle fibers. That is what is mentioned. The centromere that are replicated from the interface move away towards the opposite poles, and as a result, the motor proteins they are the ones which help in driving the centromere, and as a result, the centromere associated motor proteins they track the centromere movement and formation of spindles during the chromatin separation. And the motor proteins they convert chemical energy into ATP to form some mechanical energy, and as a result, the movement of the microtubules can take place in a very smooth way. This helps in the centromeres to move the towards the opposite poles. So the centrosomes they are splitting to form both the centrome both the north pole as well as the south pole of the cell, and over there the release of the microtubules is actually occurring. Next is the different stages of the cell cycle. The G0 phase or the resting phase is a period in the cell cycle in which cells exist in a quiescent stage, or G0 phase is viewed as either an extended G1 phase or where the cells are not at all dividing. So cell is basically in a resting phase in the G0 phase. But in case of some of the cells like some of the cells of muscle cells or liver cells or the neurons they are in the Z0 phase because they have lost their capability of dividing. Even the mature RBCs they also move towards the G0 phase because they do not need to divide anymore. Skeletal muscle cells, our skeletal system is not at all growing after a certain age when we have attained maturity. So these are the cells which do not undergo normal mitotic division, and they are in resting stage or quiescent stage, that is known as the G0 phase. G1 phase is the stage in which there is initial growth. We can see that the cell it is having lot of metabolic activity going on inside it. It means the nucleotides, ATP molecules, amino acids, they all are getting formed. Mitochondria and chloroplast are getting multiplied. Like duplication of these structures are taking place. Endoplasmic reticulum, Golgi apparatus, different kinds of RNAs, ribosomes, lipids, proteins, they all are getting synthesized. Here, when the synthesis is taking place, it is including every kind of protein which is present inside a cell, and all those proteins they are getting duplicated. So generally, G1 phase is a phase in which most of the cellular organelles or the cytoplasmic content is getting duplicated. Next phase is the S phase, where the synthesis of the new DNA actually occurs. So it is a phase in which when we say that there is presence of a single in the telophase, at the end of the telophase we have this one single uh, mm, sister chromatid along with a centromere being present. So we in the S phase is basically what is happening is it is making its another sister chromatid. Just this is taking place, and this is only known as the synthesis phase or um, replication phase of DNA. Replication is occurring. One half of the DNA is already there, another half is getting synthesized. So generally, when the cells what do the cells do in the S phase is they are just synthesizing their DNA counterparts. The sister chromatids they are getting formed. So each of the chromosome it carries a duplicated set of genes during this time. Suppose a diploid cell is there, it is not becoming it is just becoming completely diploid. Histone proteins are synthesized, and these are associated with DNA strands. Sometimes nucleosomes are also synthesized, non-histone proteins are synthesized, and this S phase is a very long phase. It is around, it takes place for 6 to 8 hours in case of normal mammalian cell. And when we talk about the first phase, that is generally the entire interface, the interface of the entire interface, it takes around 20 hours. And one cell when it is intending to divide, it can divide within a day, like one day. 24 hours is required for a cell, each cell to actually divide. So this is just a general idea. So in case of a yeast cell, it is taking place, one division is taking place, is one cell is dividing to two cells in around just few minutes. Whereas in case of mammalian cell, it's taking one day for one cell to become two. So when it is interphase, in case of interphase, the longest phase is said to be this S phase. It takes around 7 to 8 hours. Next phase is the G2 phase. G2 phase is the period when the cell machinery is getting formed, and it is the last phase of interphase. During this phase, the cell it grows, DNA it becomes replicated, all the required enzymes, which will be the cyclines or the CDKs, they all are formed during this phase. Various proteins are formed, and assembling of Golgi apparatus, certain RNAs and some proteins, centrioles, these all take place during this G2 phase. And the cell it becomes completely ready for the process of cellular division. What is the significance of the process of meiosis? Now, significance is something which is known as importance of the process. First thing is that it helps in, if you think about the process of meiosis, it is a process in which a cell is getting haploid at first, and later on it is dividing. Now, since I am discussing about the various important points or the various importance of the process of meiosis, it is important that we understand that meiosis is only taking place in the reproductive cells. Any kind of reproductive cell, it is only the one which is showing the process of meiosis. This is something which has to be kept in mind. Only the reproductive cells they are undergoing this process of meiosis, and by the process of meiosis only the gametes will be formed. So it is taking place in the sexual organs, the testis or the ova, or if you think about the plants, it's the pollen grains or the ovule over where it is taking place. Even the formation of spores is taking place the similarly in the similar way. Like some of the plants they produce spores, ascospores in case of fungi and all. If you remember, there also similar thing was taking place. Maintenance of chromosome number means that meiosis half, it just makes the chromosome number haploid. So meiosis is helpful in decreasing the chromosome number to half. If it is 2n, then it becomes n by the process of meiosis. Suppose human beings are there, so in human beings generally 46 number of chromosomes are present, but in the gamete cells there is presence of only 23 number of chromosomes. So two of the gametes, male as well as female gamete, when they combine, they become again 46, 23 plus 23, 46. But if you think about a somatic cell of human being coming in contact with another human being somatic cell, so 46 plus 46 will take place, which will be way too much than the normal chromosomal content or diploid structure of a human being. So in order to for a human being to be formed, it is very important this that this haploid number of chromosome is maintained. Introduction of variation takes place in the process of meiosis because crossing over occurs during this process. Crossing over means the maternal as well as the paternal similar chromosomes. So chromosome number 1 of mother and father, they come very close to each other, they have sticky ends and they exchange themselves. Some of the portions they exchange themselves, and as a result variation is generated. Mutation, it means that during the same process of meiosis, when new cells are getting formed, at the same time certain new characters can be developed, and these new characters which become evident, it is because of certain mutations which occur during the process of this crossing over. And not only crossing over, even during the long process of meiosis, there are certain stages where there are chances of few chromosomes getting unevenly distributed, as a result of which there can be certain um, deformities also taking place in an individual. So those can be also considered as mutations. Chromosomal mutations can occur. Evidences of basic relationship of organisms, it means that meiosis, it is something which is similar in sexually reproducing organisms. So it actually shows certain kind of similarity in organisms which are quite related. So in case of mammals, all the mammals, they are undergoing, or their sperms and ova are undergoing uh, like this haploid formation, or they are becoming haploid in the sperm cells or in the ova cells. So that is seen in all the mammals. It is irrespective of the fact that it is a human being, or it is a chimpanzee, it is a cow, or it is a buffalo, or it's any other, mammal. It is taking place in every organism which belongs to a particular category. So in this, these are some of the important significance or importances of meiosis. Next is some of the essential features of the process of meiosis. The first thing is that there is two successive divisions without DNA replication. As I have already said, that there is one division which is known as the reductional division, whereas there is another division as well which takes place, and this division is known as the equational division. So in that way there are two divisions which are occurring. One is where the chromosome number is getting haploid, and second is that same chromosome number which has become haploid, now it is also required to become single, like suppose there were 46 number of chromosomes, now 46 number of chromosomes, it has become 23. Now 23 chromosomes are there. Now these 23 chromosomes, we don't want 23 chromosomes, full-fledged chromosomes. We need 23 haploid chromosomes. So those chromosomes, they need to become half of themselves. And for that, again, we need mitosis to take place, so that there is presence of this only one chromosome or one sister chromatid. So that takes place during the second meiosis. So meiosis 1 and meiosis 2. Two phases are there. In meiosis 1, reductional division. In meiosis 2, equational division occurs. Synapsis is known as the um, it is the process in which the exchange of chromosomes actually occur. Homologous chromosomes means same chromosome of father and mother. So paternal and maternal same chromosome is known as homologous chromosomes, but they come from two parents, so that's why they are homologous. They are same chromosome but from mother and father, that's why they are homologous chromosomes. Crossing over of segments between non-sister chromatids, when I say non-sister means maternal and paternal chromatids, they come close to each other and they exchange their parts. Segregation of segregation means separation of homologous chromosomes, and then separation of sister chromatids. That's what I have said. At first equational division took place, which reduced the entire chromosome number, which was 46 to 23, and 23, when I say, so 23 full-fledged chromosomes means both the sister chromatids are there. 23 number of chromosomes will be there. Now these 23 chromosomes, they again need to get divided, so that they have just one half of a sister chromatid. So that happens during the last part, that is the um, meiosis 2 part. Different stages of meiosis with diagrams is something which is there in our syllabus, and we have to discuss it in details. The discovery of the process of meiosis took place by August Weismann in 1887, and he predicted on various theoretical grounds that the number of chromosomes must be reduced to one half during the gamete formation, so that the chromosome number of a species is maintained. And Edward Van um, Benedin demonstrated the reductional division in the same year, 1887, and the term meiosis was introduced by J.B. Farmy and Moore in 1905. These are some of the important contributions of some of the scientists for the process of meiosis. So we need to have little idea about it. We also need to know about the fact that meiosis takes place for a long period of time. It takes various, or it takes couple of days for the entire process to get completed, or we can say it takes one entire day in case of human beings to get completed. And it helps in formation of meiocytes. We have an idea what are meiocytes. Meiocytes are diploid cells, or we should say not the diploid cells, they are the haploid cells which are formed as a result of meiosis. So all the oocytes, spermatocytes, or sporocytes, they all are formed with the help of only this process, that is meiosis. I have already said that normally during this process of meiosis, it it is a process which is a very elongated process, a very long term process. So there will be various stages which will be taking place in this phase. And in this phase, it is very important that we understand that there are two things which are taking. One is meiosis 1. In the process of meiosis 1, there will be uh, reductional division which will be occurring, and it will be followed by a equational division in the meiosis 2. So this is the meiosis 1. You can very well see that prophase in mitosis, prophase 1 in meiosis, prophase 2 in meiosis has been discussed. There is a similarity between these things, that is prophase in mitosis as well as prophase 2 in meiosis. That is the reason I want you all to have a recapitulation of all the stages of mitosis, so that it becomes quite easier for you all to understand. See, in this case, during process of mitosis, in the prophase, all the sister chromatids, they are haphazardly arranged. Similarly, in prophase 2 also, the sister chromatids are haphazardly arranged. But in meiosis 1, the things which are taking place, they are quite different. There are various stages which occur during this prophase 1 of meiosis, and we have to understand this in details. This is prophase 1. Here the 2n number of chromatid or chromosome is going to become n, and that has to be understood. 2n, it means there are 46 number of chromosomes. These 46 number of chromosomes, they are getting reduced to 23 number of chromosomes in case of prophase 1. Okay, this is what we are going to understand. This is a stage which is shown over here, and Prophase 1 has various stages. We'll be studying about each of these stages. There are generally 5 stages during this process: leptotene, zygotene, pachytene, diplotene, and diakinesis. This is a short view or a overview of what we will be studying in details. At first, in leptotene, all the spindle fibers, duplicated chromosomes are formed. The homologous chromosomes, they come together and form bivalents, and pseudoepitonemal complex is formed over where synapses occurs, and as a result, crossing over occurs in the pachytene. In the diplotene stage, the sister chromatids, they not the sister, the non-homologous chromosomes, or non-sister chromat sister chromatids, they actually get separated from each other. Now they have formed, or the crossing over has been completed by the end of diplotene, and they are segregated, and as an end of diakinesis, they are completely segregated. The bivalents have moved apart and they are ready for the process of metaphase. So in metaphase, half of 23 chromosomes will go to one pole, another half of chromosomes will go to another pole. So entire chromosomal movement will be taking place, the entire chromosome will be getting segregated. Let's see the different stages. The first stage is leptotene stage. It is a stage in which it is the first stage of prophase 1 and is the shortest. It is a phase in which replicated chromosome condensation begins. So the chromosomes, they become condensed, they become more elastic, and we can very well see the structure properly. The chromosomes condense and become compact and are visible, hence making it quite possible for us to look into them and understand that they are having two sister chromatids as well. The chromosomes, they appear like strings with beads, and the beads are known as chromomeres. This is something which is a characteristic feature of leptotene. When I'm discussing these 5 points of leptotene, it is to be understood that these 5 points can be asked in the exam. Generally, a question is asked from Prophase 1 for 5 marks that describe all the stages of sub-stages of Prophase 1. So over there, you have to draw the diagrams, this diagram, which have already discussed. And then after, you have to write these few points. And the structure is as such that it has this kind of beaded structure. You can think that it looks as if certain beads, they are arranged together and they are very closely placed to one another. I'll be making two different looking chromosomes so that you can very well understand this is maternal and this is paternal chromosome, and they have beaded appearance. Each of the sister chromatids get attached to the nuclear envelopes, because in the leptotene phase, if you remember the prophase, the nuclear envelope is not completely disintegrated. So nuclear membrane will be present when we are discussing all the stages of substages of prophase 1. Next is zygotene. It is also known as zygonema. This is a phase where homologous chromosomes, they get associated closely to form pairs of chromosomes, and this process is known as synapsis. It means just pairing. So four chromosomes, they come together. You can think of four chromosomes, two paternal and sorry, I should choose, and two maternal. Like four of the chromosomes, since they are homologous chromosomes, there is centromere attached to. So they will come close to each other during this stage of zygote. The synaptic association forms up and down the chromosome, creating several points of contact, and these are zipper like structures and are also known as synaptonemal complexes. So synaptonemal complex zipper like structures are important because these are the sites at which exchange will occur later on, and synaptonemal complex formation occurs in the zygotene stage. Sometimes one mark question is asked that name the stage in which synaptonemal complex is formed. So it is the zygotene stage. After synapses of the homologous pairs, the four chromatids which are formed, they form tetrads or bivalents. Bivalents means four of them, they are very close to each other. Next is pachytene stage. This is the stage at which actually the crossing over takes place. This is a phase where the crossing over of genetic material occurs between the non-sister chromatids. Non-sister chromatids means same chromosome number 1 from mother as well as father. They are very close to each other. Now exchange is occurring during this pachytene stage. And why is the exchange occurring? Because chromosome number 1, suppose this is chromosome number 1 of father, and it this is chromosome number 1 of mother. Now the positioning of the suppose these areas, there will be same genes present at this particular loci, and that's why those genes, suppose it is capital A, small a, small a, small a, so there is a very high chance of exchanging this capital A and small a. So you can think that the entire green has little amount of red, and then again this. So this is the exchange which has occurred. I hope it is quite easier for you to understand it in this way. The synapse completes the crossing over of genetic information, and variation has taken place because entire green maternal chromosome has little tinge of red now. So that is the variation which has occurred. Separation of sister from the homologous chromosomes remains attached, hence thick complex is formed, and that is known as synaptonemal complex. The synaptonemal complex allows the formation of chiasma, which whose function is allowing crossing over of alleles with small regions of chromosomes. So I hope it is quite easier for you to understand that same loci is getting exchanged. So the similar alleles will be exchanged, and that will be creating variation. Next is the diplotene stage. In this stage, the synaptonemal complex, it completely gets disappeared, or it there is disappearance of this synaptonemal complex. The disintegration of synaptonemal complex occurs between the two chromosomal arms, causing repulsion of the arms. It means that those complex, those four two arms which are very close to each other, they are now moving back to its normal position, or they are just getting separated from each other. This causes the chromosomes to move apart from each other while still being held at the chiasma. So chiasma is the site at which crossing over has occurred, but repulsion in it means that those chromosomal arms which were not close to each other, they get separated. When I'm talking about two, suppose this was the paternal chromosome, and um, is the maternal chromosome. Now, when I'm saying repulsion, the repulsion is actually occurring in this as well as this chromosome. But chiasma formation is still there in these sites because these chromosomes, they are closely held to each other. Chiasma is formed. There is exchange of chromatin material, or alleles actually occurring during in these sites. So over here the exchange is actually occurring, and that's why they are closely held still, and that is what is written over here, that they are held together at the chiasma. Chromosomes then start to uncoil slowly, and the chiasmata has been visualized microscopically, and they can be moving close to the ends of chromatids. The process is known as terminalization. It means completely cessation of the process of crossing over. Next is the last stage of prophase 1, and that is the diakinesis. After this, metaphase will start. It takes place after the chromatids have condensed and the sister chromatids are bivalent or tetrad as visualized under the microscope. The chiasmata finally arrives at the end of the chromatid arms, and the chromosome finalizing terminalization. When I'm talking about all these stages, it is very difficult to distinguish each of the stages, that from where you'll start that the another stage has been completed. The It has to be understood in a very like basic way, that when one stage is on the verge of completion, the next stage has already started. So that's why terminalization has already started in the previous stage of diplotene. But in the diplotene, when we talk about, it just means that crossing over has completed in this stage. Okay. Diplotene is the site, is the completion of crossing over. Pachytene is the initiation of crossing over. Zygotene is initiation of synaptonemal complex. Leptotene is beaded structure formation, and the presence of non like homologous chromosomes together or close to each other, or and formation of chromomeres. This is the characteristic feature of leptotene. So this is how we will be able to discriminate each of the stages. We were in the diakinesis stage, and here you can we will just look into the fourth point, that the chromosome become condensed and remain connected by the chiasmata and cannot move further towards the poles. At this stage, the nucleolus and the nuclear envelopes dissolves, allowing the centrioles to form the microtubules, and the spindle fibers are formed during this stage because metaphase is on the verge of getting started. This metaphase is metaphase 1. In this metaphase 1, what will happen is the entire chromosome pair which is formed after crossing over, one of the chromosome will move towards one side, another entire set of chromosome will move towards another pole. In this way, 23 number of chromosomes will be there in one side, 23 in another side. So here let's just have a concised vision of how metaphase is occurring. Here you can see in prophase 1, 1, 2, 3, 4 chromosomes are there. When there is a dark blue chromosome, that is paternal. Light blue is maternal. Orange is suppose paternal, red is maternal. In this way, in metaphase, you can see that entire chromosome 1 has moved towards one pole and chromosome 2 is moving towards another pole, isn't it? But in anaphase 1, what you can see, half of, not half, the entire chromosome is moving towards one of the sides. So it is not the maternal or paternal one. In metaphase 1, you will see that placement of all the chromosomes in the central equatorial plane is taking place. But in the anaphase, you can see that one entire paternal one, suppose this is paternal, this is paternal chromosome, this is maternal chromosome, both are chromosome number 1. So chromosome 1, chromosome 1, this is chromosome 2 paternal, this is chromosome 2 maternal. You can see what is happening over here in anaphase 1. Chromosome number 1 of father is moving towards one side, chromosome 2 of father is moving towards another side. Suppose this chromosome 2 is of mother and this is of father. So in this way, what will happen, chromosome number 1, 2, 3, 4 till chromosome number 23, let me make two cells, uh, not two cells, let it be just a single big cell. What exactly happens is chromosome number 1 of father, here chromosome number 2 of father, 3, 4, suppose 23. Chromosome number 1 of mother, chromosome number 2 of mother, chromosome number 3, chromosome number 4, chromosome number 23. Now you can very well understand. See, there is no as such discrimination that father's chromosome is only going towards one of the this pole. It can be father's chromosome or it can be mother's chromosome. The entire chromosome will move towards one of the pole. So 23 chromosomes will be here in one pole and 23 in another pole. When it divides for the second time, these 23 full-fledged chromosomes, half of it will move towards one pole, another half towards another pole, and as an end product of meiosis, 4 number of haploid cells will be formed, and that is the end of meiosis 2. Here it will be ending. So anyways, we'll be dealing with the metaphase part in the next video, but here this is just a condensation of entire thing which you all have to understand. You all need to go through the textbook once in order to understand it little more well, and if there is any doubt we can discuss it.